Prof Anna Matiti, Executive Dean, Faculty of Science. The reason why we are here, Professor Lizelle Piater, the inductee, Prof Ian Dewberry, so nice to see you. The respondent, Prof Dewberry is the Director of the Research Center for Plant Metabolomics. Senior leaders of the University of Johannesburg, members of Senate and other academics, distinguished guests, of course many students, all who have joined us here both in person and online. Sanibonani, goeienaand, good evening to Bella. It is indeed a great honor, Lizelle, it is. It is indeed a great honor and a special privilege for me to welcome you to the Professorial Inaugural Lecture of Professor Lizelle Piater. As I do so, I wish to express a warm welcome to her loved ones, special guests, and to her colleagues. It is indeed a proud and a joyful moment for us, for you, but it is also a landmark moment for all of us here, for higher education in South Africa and even beyond. The inauguration of professors is a public ceremony, which is why we are also live streaming this event currently. And then during this event, newly appointed professors are inducted into the office by the Vice-Chancellor or the Vice-Chancellor functionary to deliver their inaugural addresses. The ceremony has its roots in the medieval university, and it serves several purposes. First, it is an expression of welcome and entry for new professors joining the circle of colleagues who are already professors. Secondly, it also provides a platform for professors, for you, Lizelle, to display your expertise in your specific discipline and to showcase your research. In the colonial tradition subscribed to by many universities, assuming the role of the professor actually refers to the gown and the cap, and this is what we see here. Once we've listened to your inaugural address, the gown, which really denotes the professorship, will be formally placed over your shoulders. Today, ladies and gentlemen, friends and colleagues, we gather to witness Professor Piater's entry into this illustrious community of scholars at our university. It celebrates her contributions to the discipline and ultimately her impact on society, because that is what we do, that is what we stand for, to make an impact on society. Professors provide a university with its identity, with its character, with its academic legitimacy, and I think perhaps most importantly, with its integrity. The inaugural lecture is a rite of passage, following the confirmation of the appointment of a person as a professor. And this evening, we will listen to Professor Lizelle Piater as the gown goes to town. And by this, I mean that the power of the inaugural address is when the expertise of the inductee is showcased beyond the corridors of the university. And these expertise then reverberates with society. It stands out as a moment of pride for the incumbent, the family, fellow scholars, the university, and ultimately as a moment of pride for society. In the past, several philosophers and scholars have defined universities using fairly narrow conceptualizations. For example, a search for truth, for universal knowledge, and even as instruments serving the purpose of the economy or being utilitarian in nature. Through our work and through our research at UJ and through your work and your research, Lizelle, I hope that we can break out of these fairly narrow conceptualizations and reflect on the university as contributing to the public good, working towards societal impact. What then is the role of the intellectual? And I think this is quite, an important question that we all grapple with. Edward said in one of his articles, defined the role of the intellectual as one who commands a vast knowledge of a specific discipline. Lizelle, that's you. One who is rigorous in their analysis of the literature. Lizelle, I think that jacket also fits you. One who views being an intellectual as a vocation. I know for a fact that's you. And who functions as a kind of public memory to recall what is forgotten or ignored, to make the connections that otherwise are hidden, and to provide alternatives for mistaken policies. It remains then for us, as a university with a Pan-African vision, to derive our mandate as intellectuals and as professors. And importantly, how do we embrace the role of a professor as a disruptor while continuing to be flagship carriers of our discipline? This evening we will listen to Professor Lizelle Piater as one further step, only one further step in her journey of being a professor. This journey, which does not conclude, it didn't begin with this lecture, and it certainly will not conclude with this lecture once this lecture has been given. Notwithstanding, this inaugural lecture does create an opportunity for self-reflective pause in the journey of being a professor. Let me now invite the Executive Dean, Professor Anna Mutiti, to introduce Lizelle Piater.
Good evening. Good evening. Dumelang. Madam VC Functionary, Professor Van Vieren, please allow me to observe protocol. Ladies and gentlemen, it gives me great pleasure to narrate the CV of Professor Lizelle and Piater. Lizelle was born and raised in the small town of Kroonstad in the Free State. Following matriculation, she pursued a BSc degree at the University of the Free State, majoring in biochemistry and chemistry. Subsequently, Lizelle completed a BSc Honours in Biochemistry with distinction, followed by graduation of a master's degree in classical biochemistry under the supervision of Professor Derek Lithua in 1998. She then joined the group of Professor Ian Duberry at the then Rand Afrikaans University and now University of Johannesburg in the field of plant pathogen interactions, focusing on lipopolysaccharides known as microbe-associated molecular patterns, or MAMPs. In 2003, Lizelle continued research in the same field as a postdoctoral research fellow in the lab of Professor Neuberger at the Leibniz Institute for Plant Biochemistry in Halle, Germany. In 2004, Lizelle took up a position as lecturer in the Department of Biochemistry and Microbiology at the University of Free State and joined the extremophiles group. Subsequently, she was appointed as senior lecturer in the Department of Biochemistry, University of Johannesburg in 2009. Again, continuing the field of plant pathogen interactions, focusing mainly on proteomics and later metabolomics. Given the proteomics work, Lizelle was contacted by SA Venom Suppliers in 2021, and since then has shifted her focus to the field of snake and scorpion venomics, with the aim of improving the production of antivenoms as well as application of toxins for use in medical treatments. To date, excuse me, Lizelle has published over 85 publications. She is currently C2 rated by the National Research Foundation and has a Scopus H index of 26. She raced through the ranks at UJ to full professor in 2022 and is currently the head of the Department of Biochemistry. Lizelle is happily married to Professor Reinhard Mabel, a full professor in chemistry, and they have 2 Neapolitan Mastiffs that keep the house very busy. In her spare time, she enjoys folding origami. Ladies and gentlemen, I give you Professor Lizelle Piater.
Thank you, VC functionary, Madam Dean, colleagues, friends, and family. So, a title that will probably raise eyebrows for many and means different things to different people. But in biochemistry, promiscuity is a perfectly accepted term. In fact, evolutionary biochemists prefer to reserve the term promiscuous to refer to interactions that are not physiologically related. And as I walk you through my journey of research, it will become apparent that as I've evolved in my research fields of interest, often unrelated, but still pulling through the golden thread of biochemistry. So I started off my MSc in the classical field of biochemistry with the purification, characterization, and application of a fungal tannase under the supervision of Professor Derek Littauer. Now, ingestion of green tea often leads to hepatotoxicity or damage to the liver, and this is often due to this compound, which is epigallocatechin 3-ol-gallate. But once hydrolyzed to epigallocatechin, this has more health benefits on the human body, and has even been shown to have increased free radical scavenging activity. So in my project, we were able to purify and characterize this protein, and we were able to show that this compound can be reduced, importantly with retention of stereochemistry, which at that time chemistry could not achieve. Now, unfortunately, I am unable to show you the purification and characterization because all this data is still on one of those old floppy drives that we had way back in the day. But I can tell a story. So to do the characterization, we had this very fancy, in those days, Beckman spectrophotometer that had the inlet to which you could couple a to which you could couple a circulating water bath. And colleagues of my age, roundabout and above, will agree that it was quite a step up from that which we were trained in the undergraduate, where students would, or one of the students would have to shout time, and somebody would have to try and figure out what exactly number the dial is on. Nonetheless, during my characterization studies, the circulating water bath tube sprung off, and I flooded the motherboard. And for that, at the end of the year, I non-proudly received the Titanic Award. I then moved into the group of Professor Ian Dewberry, particularly in plant pathogen interactions, and focusing on lipopolysaccharides, or LPS, as a microbe-associated molecular pattern, or a MAP. Now, for those of you who might know in the medical field, LPS is also what is responsible for causing septic shock in humans. So it was interesting for us to see how plants perceive LPS and how they mount a defense response. In this project, we initially showed the rapid and transient phosphorylation of tyrosine, and we later narrowed that down to be a MAP kinase using a specific antibody and immunodetection. The rapid activation was optimal at 7 minutes, and 100 micrograms per milliliter LPS. And when we treated the system with an upstream MAP kinase, we completely reduced the phosphorylation, indicating that this protein is indeed involved in the perception of LPS. In-gel kinase assays displayed a protein with a molecular mass of 43 kilodalton and a PI of 6.1, while two-dimensional gel electrophoresis indicated two charged isomers of the MAP kinase, and this work was the first of such a MAP kinase reported in plants. Thereafter, I moved to do my postdoctoral research at the Leibniz Institute for Pflanzenbiochemie in Germany, and this was in the city of Halle, which is the birthplace of Handel. And as you will note from the map, Halle is quite far east, and you did very well there if you could speak Russian or German. So I quickly had to enroll into German classes. My host was Professor Thorsten Nürnberger, and when he sat me down to explain the project, he spoke of terms like PCR, RT-PCR, cDNA cloning, expression vectors, and I thought, is this man speaking German? Because I had absolutely no background in molecular biology. What I knew about molecular biology was the first PCR machine I ever saw, which was the one that you would manually move your tubes in comparison to the ones we had today. Nonetheless, so I delved into the project, and prior studies had identified 20 genes of interest from microarrays. I then had to verify those 20 genes using RT-PCR, and for these 20 genes, I then continued to order 47 knockout lines, in other words, where the genes are deleted, and I had to propagate these seeds and verify the lines to truly be knockouts using PCR. We also biochemically characterized these knockout lines using infection studies, rating symptoms, looking at the hypersensitive response, as well as the PR gene expression. From that, I constructed full-length cDNA clones into gateway vectors, and by the time the lab moved to Tübingen in October of the same year, and I moved back to South Africa, I had characterized 5 full-length clones and 5 kinase domains. But I had a side hustle project. So because of my interest in LPS, I also continuously delved into the literature of what is known about LPS in mammalian research. And I continuously looked for clues or genes or proteins that were identical to those possibly in plants. And I draw your attention to these 2 proteins highlighted because they will feature later. You can see the date of the file was 2003, so I'm not talking nonsense. I investigated as much as I could, designed primers, indicated knockout lines that we could use, and went to my host with this idea. And he just shut it down and said, no, he does not think it is viable. But nonetheless, I would like to thank these string of people who were part of the research group, and we were also invited by immunological reviews to submit a manuscript of striking similarities and obvious differences between the innate immunity in plants and animals. I then joined the group of the extremophiles. Now, the idea of the extremophile groups was that we would sample extreme environments or contaminated environments, that we would sample as much water as we could, that we would use vials with specific media in to try to enrich for specialized cultures, and we also sampled these masses of slime, which is really micro, or at least biofilm of bacteria stuck together, which the Americans termed snotites. And from the micrographs or the microscope slides, you are able to see the wide variety of shapes of different bacteria within these samples. One particular trip, this one, was extremely challenging. And Esther helped me out, I think it was Tautona, which was 3 kilometers down and a 3-kilometer walk in with all of our sampling equipment. The guys had just hit a water pocket, which they sealed off. We went in, it was extremely hot and humid, and the water temperature sampling was at least about 65 degrees Celsius. And we had 3 men and 3 women, and halfway through the sampling trip, all the men overheated. In fact, the one thought he was seeing mice running around, and when Prof Derek Litauer lifted his helmet off his head, steam just came off of his head, which left us 3 ladies, Esther, myself, and Elsabe, to complete the sampling trip. And which is actually very interesting is that nowadays there are more lady miners because women actually have higher heat shock proteins than males, and especially during their menstrual cycles. So we would go back to the labs then, and we would again couple the water onto enrichments. Most of the work was conducted in anaerobic chambers because these organisms had not prior been exposed to oxygen. And this is just an indication of a picture showing how many bacteria there can be on the tip of a pin. Then in 2007, the metagenomics platform was formed, and this was funded by the then Piater, who is now TIA, to the tune of 13.7 million rand. The idea was that we would sample extreme environments, that we would look for specialized cultures, extract DNA, prepare cDNA, eventually put these into libraries, and then go through screening process for eventual biological applications. And just to give you an idea of the diversity of these libraries, that over 40% of the bacteria identified, or at least were, the diversity studies indicated that over 40% were unidentified, uncultured bacteria. And this was one of the first papers, which I just briefly indicate, of the mines, where you can see in bold how many uncultured bacterium there was, and what a vast variety of material we had to work with. During that time, there was an outbreak of typhoid fever in Limpopo, and it was unclear whether the cause was Salmonella typhi, which causes death, or Salmonella typhimurium, which causes gastroenteritis. So off we went, sampled the water, came back, and then we compared molecular techniques to those of conventional, time-consuming microbial identification techniques. And we could clearly show that the molecular techniques were far superior and quicker to those of the conventional methods, and that the causative agent of the typhoid outbreak was, in fact, the Salmonella typhimurium. We were also able to isolate a copper-resistant mine isolate. This specific organism was identified as Proteus mirabilis, and it showed a minimum inhibitory concentration 3 times higher than that already reported in literature. And we were able to narrow down the gene which is responsible for the resistance in comparison to the controls. Apart from that, there was an organism called Thermus scocoductus, SA01, who showed excellent chromium reduction activity. Now, chromium-6 is known to be toxic and carcinogenic, in other words, causing cancer, while chrome-3 is known to be innocuous or harmless. Now, it was also interesting that this enzyme was identified as one of the family of an old yellow enzyme, which is actually an NADPH dehydrogenase that is responsible for the reduction of activated alkenes, and again shows the promiscuity of proteins. In the same organism, a thioreductin reductase was identified and showed excellent specific activity when compared to a known soluble ferric reductase. Also important is that as there is the interconversion between iron 3 and iron 2, toxic free radicals are generated and can be of detriment to a specific organism. And it makes sense that this organism would have this enzyme, because what bacteria do is they release cytophores into the environment where they pick up or chelate the iron 3, bring them back into the cell, and then these reductases reduce it to iron 2, which they can then use as a micronutrient. But the most interesting project was from the same organism. There was the ability to reduce uranium-6. Now, uranium-6 is insoluble and highly toxic. And when converted to uranium-4, this is soluble and can easily be remediated or cleaned up. This organism was able to easily grow on uranium-3, and no matter the growth phase or the electron donor, it was able to reduce the uranium-6. We used EDS, or at least 10 micrographs, to show that this uranium is in fact precipitated on the surface of the bacteria, and this was confirmed by, and this is really very small, by an EDS spectrum to be uranium in comparison to the untreated cells. When homology modeling was done, it was found that there was a surface exposed disulfide bonds that could be attributed to the reduction activity. And this was true because when cysteine residues within this area was mutated, the enzyme completely lost its ability to reduce uranium. Importantly, again, this protein was identified as an ABC transporter, which again indicated the promiscuity of proteins. This work also resulted in 2 patents. Okay, let me just have a sip of water. After I left, the colleagues discovered a new nematode or worm in these mines, which they called Halicephalobus mephisto. And just to say, Mephisto is one of the chief demons in German folklore. And this organism, and of course this was published in Nature, very proud of you, Esther. This organism was discovered in 3.6 meter deep fracture water, and carbon-14 data indicated that the fracture water was between 3,000 to 12,000 years old. And you may ask, what does this nematode feed on? The exact same bacteria that we were sampling. Thereafter, my colleague, Esther, established her own company, iWater, in which she currently looks at the bioremediation of contaminated water as well as contaminated sites. So then I moved back to my roots, pun intended, plant pathogen interactions. And the first of the studies was using LPS again and labeling it firstly with a conventional fluorophore as well as quantum dots. And the scientific colleagues should know that last week the 3 gentlemen who discovered and synthesized quantum dots were awarded the Nobel Prize in Chemistry. So we were able to convincingly show using protoplasts that there was an increase in fluorescence with an increase in concentration, so it was concentration dependent. It was also time dependent, and as can be seen in these 10 micrographs, where the fluorescence of the normal plasmid up to the complete green. The more protoplasts we put in, the higher the fluorescence was. We also showed that if we pre-treated the protoplasts with unlabeled LPS, we significantly decreased the fluorescence, and this indicated the reversibility of the binding. We were also able to show, using endo and exocytosis inhibitors, that the fluorescent was significantly decreased, and this indicated that the receptor or binding site would flip-flop within the membrane as it took LPS into the cell and recycled itself for further uptake. With that in mind, the question was always, LPS that we worked with is termed smooth and has 3 moieties: the lipid A moiety, the core, and the O-chain O-chain moiety. And it was never clear if the entire molecule or if one of the moieties are required for the perception and recognition. So we then chose to use 3 matrices to immobilize the LPS, and that would mean that each of these matrices would capture the LPS on one of these moieties, exposing the other 2. We then washed over plasma membranes, washed off any unbound proteins, and eluted those that had bound to the LPS. We identified a large number of proteins, and I won't bore you with that, but I want to highlight out BAC1. Now, BAC1 is known to be the co-receptor of a protein, FLS2, which is responsible for the perception of flagellin. And flagellin are those little tails at the end of bacteria that allow them mobility. We also identified a myrosinase, which is a glycan-binding protein, hinting towards that possibly the polysaccharides in the LPS is essential for recognition. In a parallel study, we did exactly the same, except we did not label the LPS. We used 1D and 2D electrophoresis, where we cut out bands that were prominent in the treated samples or spots that were specific in the treated samples, and we also cut out entire lanes and sent those away for identification. We were able to identify 95 proteins from all of the protein samples, bearing in mind that each of those lanes had about 1,800 proteins. And again, I highlight out the fact that BAC1 was identified in 2 or only the treated samples. Then our bubble was burst. A paper came out in Nature Immunology in 2015 from this group who claimed that they had in fact identified the LPS receptor, which they termed LAW. Unbeknown to us at the time, this claim would be refuted in 2019 when it actually showed that LAW recognized medium-chain fatty acids, which were probably contaminants within their LPS preparations. But nonetheless, we didn't know that at the time, and the show had to go on. So we turned our focus to ergosterol as a fungal MAMP, and we coined it an orphan MAMP because of the little literature that was available in the field. We also proposed a mechanism for the perception of this ergosterol. So in the project, again, we used the plasma membrane, we used ergosterol, and we immobilized that on 2 different matrices. We ran over the plasma membrane, and again, we identified a large number of proteins that I will not bore you with, but interestingly, proteins that were also identified in the LPS perception and chitin perception. And that always raised the question as to whether each MAMP has an individual receptor or binding site, or whether there was promiscuity in the recognition thereof. We, in a separate study, while there was an ongoing metabolites project, looked at the proteome analysis of Nicotiana tabacum, or tobacco cells, following treatment with isonitrosoacetophenone, or INAP, which acted as a priming agent. Again, we used 2D electrophoresis, but we also used eye-track analysis, and we were able to identify 125 proteins from Nikita. I don't know how many thousands you went through, and we were able to categorize these, and we were able to discuss them in terms of the defensive state that ergosterol induced, and we were able to also correlate of that data with the metabolomics data. Then, now that we knew the LPS receptor was still out there. We returned back to investigating the receptor or the binding. We used 2 LPS chemotypes. The first that we termed the wild type, and the second that was a mutant, and it was mutated in its core and in the O chain. We again used the same matrices to immobilize the LPS, and again from the Venn diagrams you can see numerous proteins that were common, but also proteins that were distinct for each of these matrices. Again, we identified a myrosinase, but I draw your attention to the fact that we identified this LBP family protein. During that same time, 2 manuscripts came out which indicated that LBP bound directly to LPS and induced a defense response in plants. And now you guys will remember this was my side hustle in my PhD. So then we moved on to design a very ambitious PhD project, I'm sorry Ben, in which we looked at both the metabolomics and proteomics of 2 knockout lines. So the back one that we had identified successive times, and then the LB2. And we started off by first reviewing the prospect of using gene knockouts in functional studies of innate immunity, and we proposed in a figure what might happen when these binding sites or receptors are not present for the particular MAMP. So from the proteomics leg of the project, we were able to identify common but also distinct proteins when comparing the wild type to the 2 knockout lines, and in particular when using the 2 different chemotypes. Also of importance was that this work hinted towards the, what's the word I'm looking for? That LBP2 and BAC1 were possibly involved in the perception of the LBP, LPS, and was determined by the moieties of the 2 chemotypes. When we categorized the proteins, again, we found that proteins we identified had previously been identified in other bacterial MAMPs and fungal MAMPs signaling. And that then posed the question whether in fact there is more than one receptor or co-receptor for LPS, and again indicating promiscuity of enzymes. The network protein-protein analysis showed clear distinctions between the knockouts and the wild type, depending on which of the 2 chemotypes were employed. And this again gave us an indication that the 2 selected proteins were possibly involved in the perception and signaling. From the metabolomics data, 45 significant metabolites were annotated, and these depended on, again, the 2 chemotypes and the genetic background of the plants that were used, and were categorized into the various categories. The distinct metabolite accumulation was again dependent on particularly the chemotypes, and hinted again towards the possible role that LBP2 and BAC1 will play. And just in summary, so of the significant proteins and metabolites that were identified following treatments with the 2 chemotypes and the various genetic backgrounds, these were then summarized and reported with regards to comparing the wild type to the knockouts. Right, then on to new ventures. So due to our proteomics knowledge in the group, we were approached by SA Venom suppliers who was very interested in profiling the different venoms within genus and between species. And the reason being that you might note of late, there is a scaringly scarcity in antivenom, and the production of antivenom is extremely expensive. It's usually between 2,500 Rand to 5,000 Rand per vial, and could require between 10 and 15 vials per patient. And also, the antivenom is not always specific enough. So this project aims to obtaining the venom. Luckily, we obtain it in a powdered form, and I won't go near that form anyway. You then do usually 1D and 2D electrophoresis, then reverse phase chromatography to fractionate. Again, proteomic analysis, digestion of the proteins for subsequent protein identification, and then to use this knowledge in the production of antivenoms and for other therapies. So it will be interesting for you to note that although venoms are toxic, there are numerous FDA-approved drugs on the market that are used in humans currently and that are derived from snake venom. And our other therapeutics will include, due to research in our department, anti-cancer therapeutics. So we are still very much in the early stages of this project. And just as an example, these are a couple of different species from the same genus, the Naja genus, and you can clearly see the vast difference in the proteomic profiles, which would of course influence then the antivenom production. Also, very clear from the 2D analysis. When we do proteomic, at least biochemical assays, we note that the orange and gray are really the antivenoms, and you will note that in 3 from the same species, how very little efficacy this antivenom has on sample number 3, and hence the need for new broader spectrum antivenoms. We are currently in the fractionation stage for the peptide identification, after which we will then do our studies on cancer cell lines. Also, the supplier has of late become interested in venom or scorpion venoms. And just to give you an idea, this is a profile of a South African species in comparison to a North African species. And again, you can see the differences between them, although they belong to the same family, as well as from the 2D. As I said, the project is in its infant stage, but Ben has to date published 2 review articles, one on profiling specifically African snake venoms, and one on the potential of the snake venom toxins as anti-cancer therapeutics. And with that, colleagues, I move on to my gratitude. The first being Dr. Esther van Heerden, who is in the audience. She was a PhD and my mentor in my MSc when I started, and I was quite afraid of her because the first time she showed me what to do in the lab, she said to me very harshly, there are 2 ways of doing things, the right way and the wrong way, and I'm going to teach you the right way. And I hope that I've conveyed that to my students as well. And subsequent to that, she has become the Michelle of my Romy and Michelle movie. Professor Derek Littauer, who, when you would print out your purification profile and you would take it to him, he would hold the paper horizontally and say to you, your buffer was too cold. Then Professor Dewberry, who I've now been with for many, many years, I always tell the students, if you need to know something, go to Professor Dewberry, because his knowledge far surpasses anything you will find in a textbook or even in literature. Gratitude to the colleagues in the Research Center for Plant Metabolomics, who I could co-supervise, and then, of course, especially to our group, the Ubuntu group, without whom I wouldn't be where I am now, and we are not just called the Ubuntu group for no reason. My students quite readily lend out reagents to various research groups, which we don't often get back. My gratitude to my colleagues for their collegiality, their camaraderie, and their support. And with that, we were afforded the Top Team Award in 2022. My good friends, I'm not going to call them by their titles, but rather Fluffy, Heather, and Jacinda, for keeping me sane and for actually answering all of my emails. To my long friend Gerrit Koosen, who I've known since his second year when I was in my M, and I actually demoed for him. My current deputies, Fid and Msisi, for making us a strong management team to run the department smoothly. And a word also goes out to their predecessors, Fluffy and Gerrit, who also acted as deputies. My besties, Marianne and Marisha, for all your love, friendship, and support, and for much needed girls' weekends away, or at least an annual weekends girl away. Marisha specifically had a cup designed for us, which we take on our trip, and this mug not only serves as coffee in the morning, but also as adult beverages in the afternoon. My home family, Nkazi, Yami, Gogo, and Olu, and especially Nkazi for being with us for many years and for walking with us through the good times and the sad times. Thank you for all your love. My gratitude goes out to also my family in the Netherlands who are far, far away and are probably watching online. Hoi familie, heel veel liefs en knuffels vir julle. And a special word goes to my father-in-law, Pa Karel, who actually was prepared to fly out for tonight, but whom we could not accommodate to our very busy October schedule. So he will fly out later in the year, but for all his messages throughout the week of encouragement. My mother-in-law, Mariki. Now, I know quite often when people refer to mother-in-laws, they roll their eyes. That is not me. My mother-in-law sadly passed away earlier in the year, but she was a wonderful woman. She was warm, and she volunteered freely to help on helplines for people with medical conditions and suicide attempts. You will be missed. To my second parents, Chrissy, Tani Sandra, thank you that I can call you at any time for any advice. Thank you that you are always there for me, that you support me, and that you love me. My three sisters from another mister, or another misters at least, Lynn, Malit, and Kari. Thank you for all your love and support, phone calls, WhatsApps, and for being the sisters that I never had. Okay, then of course our children who have or give our house a lot of fun, love, happiness, but often disasters. Those that have passed but leave deep footprints or paw prints in our hearts, and then the current two hooligans who have cost me 3 pillows, 2 pairs of slippers, 3 slip-slops, and a feather-down duvet within the case of 3 weeks. Then my parents, my mom and my dada. Thank you for all your love, for all your support, and may I just say they have both passed away. Thank you for affording me the education to be where I am today. Thank you for loving me unconditionally, even when I came home with my blonde hair dyed red, even when I came home with braids in my hair, when my father saw my tattoo for the first time. And I must just add, my family all call me Zali or Zal, and when my father called me on my first name, you must know there's trouble. So when he saw the tattoo, he said, Lizelle, is dit 'n tatoeëermerk? And I was already a working woman at the time, but I knew I was nonetheless in trouble. when I brought home a boyfriend with long hair, that my father had always said, if you do so, I will bring out the weed eater. Well, Reinhard's hair survived. My father always used to say to my mother, my vrou, ek weet nie waar kom hierdie kind vandaan nie. And to translate it into English, my wife, I really don't know where this child comes from. And then last but certainly not least, my husband and partner of 17 years, for all your love and support, for debating with me, for loving me unconditionally, for educating me. Like yesterday, I was completely confused about who the Hamas and what's the other group? Hezbollah was. And just for being my best friend and the love of my life. I thank you. I thank you all.
Prof Piater van Vieren, Registrar and VC Functionary, Prof Anna Mutete, Executive Dean of our faculty, members of Senate, colleagues, students and honored guests, both present and online. It is a great privilege for me to act as respondent for Lizelle Piater. I've known her since 1999, acted as supervisor of her PhD studies, and created the opportunity for her to go to a leading international laboratory in Germany for her postdoctoral studies and training. Upon her return to South Africa, and specifically to UJ in 1929, we continued our collaboration, which has thus far resulted in about 75 papers during the last 14 years. But apart from her excellent presentation that you listened to tonight, who is Lizelle Piater? First of all, she is a multi-talented person. She is not only a good scientist and researcher, but also has the ability to share that knowledge, which makes her a good teacher, lecturer, and she's able to constructively motivate her students. To that, we can add a caring and compassionate person, a good leader that supports her colleagues, and that creates an environment in which they can excel in return. She is a member of staff who can be depended upon to go that extra mile, and in my view, she embodies the core values of UJ, and she's an exceptional asset for this university. Right, so as part of her presentation, Prof Piater shared with us her journey as a biochemist, from a doctoral student to a PhD graduate to a postdoctoral fellow, and eventually transition into academia. Now, this was her specific journey, but fellow biochemists might have followed a different journey, because our common field of expertise, biochemistry, is indeed a promiscuous subject, as she rightly pointed out. Yes, the term promiscuous can mean different things to different scientists. In her case, biochemistry, a specific biochemistry background, was established when entomology and metabolism were the two main subdivisions or focus areas within biochemistry. And perhaps they still are the two most important ones. But anyway, at that time, the concept of substrate specificity, meaning that enzymes have unique substrates and create a unique product, that was quite paramount in biochemistry teaching. But as the subject evolved, we came to realize that this is not necessarily so, and that some enzymes can act on more than one substrate, and one enzyme can potentially create more than one product. So we came to understand that there might or already is some level of promiscuity at the level of enzymatic catalysis. So going on a few years into that point, with the advent of the ability to manipulate nucleic acid polymers, it is the DNA and RNA within cells, the subdivision of molecular biology was born. And this led to an explosion in biochemistry knowledge, and the so-called omics fields were soon followed. With the omics fields, I mean genomics, transcriptomics, proteomics that Lizelle mentioned frequently, and also metabolomics. Now, these led to more promiscuity in the broader subject of biochemistry. And these omics subdivisions rapidly became the new buzzwords in biochemistry, and we as biochemists had to reinvent ourselves to stay current with these new ways that research in the life sciences was to be conducted. And this was also apparent in the research outputs of Prof Piater, and one can say that she has lived and hopefully enjoyed the journey thus far. Right, with regards to the way forward, I do want to stress that we as scientists cannot afford to stagnate, that we have to stay relevant in the pursuit and the generation of new knowledge. And to highlight this point, I want to share a few lines from a book by Dr. Craig Venter, who was sometimes a controversial scientist during his long career. And this concerns his ocean exploration genome projects that can be described as metagenomics, which Lizelle also pointed out. Now, the name of this book is The Voyage of Sorcerer II, and this book was only published in September last month. Now, the Sorcerer II refers to the name of the yacht that belonged to Dr. Craig Venter, and that he used to sample ocean waters from across the world. But in the book, he describes the outcome of the project, and I want to quote, it's beautifully written. The result of this project was a massive library of millions of unknown genes, thousands of unseen protein families, and new lineages of bacteria that revealed the exquisite biodiversity and the unimaginable complexity of life on Earth. Craig Venter then continues in the book on how we as biochemists can harness this microbial genomes to develop alternative sources of energy, food, and medicine. And I think it's very apt, given the current conditions and the focus on sustainable development, sustainable utilization of the planet Earth. So with these responding remarks, I would then like to end and, in my personal capacity, welcome Prof Piater to the cohort of full professors at UJ, and by extension also to the cohort of biochemistry professors in South Africa. Congratulations, Lizelle. We really enjoyed your presentation, and as I said, welcome.
Reinhard, would you like to do the honors?
I did not expect that.
It is not an easy thing to put this gown on. I can tell you, you've done it perfectly. Everybody in? Thank you. Okay, I have to start by saying that I have benefited from the Ubuntu Lab. I just need to remember what I benefited. Okay, Lizelle, you know, I never really watched Big Bang Theory. I remember on one of the Voyager's research trips down to Marion Island, it's about 5 or 6 days on the SAO Gallows. You know, I was so focused on lying in my bed, feeling very poorly, and lots of people were then binge watching Big Bang Theory, and I didn't know what they were talking about, and that was years ago. In any case, and then, you know, when my feet hit sort of vasteland, you know, terra firma, I actually picked it up. And one of the things is that I have in common with Sheldon is that when I'm in turmoil, I always defer to tea. And you have just burst my tea bubble. So tomorrow I will need to find something else to help me deal with the turmoil in my life. But on a positive, from your presentation, I also did note down a few passwords. So the half of my turmoil comes from when I have to think of new passwords, but you fixed that, so I did write down a few words that I can use as passwords. Then I'm also sort of quite excited that there are people in the audience that know what a floppy drive is, because nowadays it's not an easy thing, you know, to explain to students what it is. I also remember PCR machines being boxes with light bulbs, you know, that you had to sort of randomly switch on and off. I don't know if you also, but I always had to nervously wait for the Geiger lady to come and inspect the lab. You know, that was like terrifying in any case. You know what, Lizelle, one of the things when I was studying that my aunt said to me is, you know, when I tried to tell them what my projects were on, they always said, so what? And then I sort of dreaded that question because I never knew what would I say. You know, the more I tried to explain, so what, the more they looked at me and said, so what? And you have perfectly explained your research and the so what of your research. And I think that is critically important. So, you know, whilst I often dreaded so what, I think you welcomed it. You know, you just, you know, so what was sort of part of your work. And that so what is actually critically important for all of our research. You know, if we can't answer so what, you know, what is the point then? And you answered a lot of so whats this evening, and for that I absolutely salute you. It also shows that your work has relevance to several aspects of society, and I think that is critically important. Lizelle, I've known you for many years. In fact, I must admit that you are my, well, are still, but you know, when I was in science full-time, that you were my go-to person. You were always my go-to person. You know, you would be the one that says, let's apply for, and it doesn't matter what it is, you know, tomorrow or during the night I would have to write an application for an NGAP or a whatever because Lizelle said, come on, you know, let's apply for. And that was your motto, you know, let's apply for, let's make the most of every opportunity that comes our way. You know, you were always the first one, and you are still, I'm sure, the first one as an HOD to have all of the tasks doesn't matter. You know, we're still trying to work out what does performance management mean, or, you know, how do you do this? And Lizelle had already done it and was explaining to all of us how to do it. You know, your general outlook, I think, on everything in life is let's do it. If not, why not? And I absolutely appreciate that. I also remember our Jerusalem challenge, you know, which was amazing. And, you know, you are that glue that holds together not only, I think, your department, but you don't know it, but you hold together a lot of the faculty, just by being who you are. And for that, and your research, I salute you, and Yuja applauds you. And we are very, very lucky to have you, Lizelle. Okay, ladies and gentlemen, I'm very aware that I'm standing between you and a glass of champagne and something to eat. A shout out to our School of Tourism and Hospitality. Many of the people that you will see are actually students in the school. So just a big thank you to them. They absolutely always stand ready to cater for us. And then last thing to say is, well done, Lizelle. Thank you to everybody that have joined us, both here and online. Lizelle and I look forward to the next, I don't know what to say, 50 years, 40 years, 30 years working with you. Thank you very much. Thank you, ladies and gentlemen.
The University of Johannesburg, the future reimagined.